everyone. We're going to get started in just a minute. Before we begin, I just wanted to go over a really quick disclaimer. I just wanted to remind you guys that we do post these videos on a public forum. So you may wish to remove your name, your last name.
Right, everyone. We're going to get started in just a minute. Before we begin, I just wanted to go over a really quick disclaimer. I just want
Sorry about that. Okay. As I was saying, we're going to post these videos to YouTube, so you might want to just take your profile picture off or remove your last name. And just continuing on this lecture means that you do understand the disclaimer and that you consent to it. Can everybody hear me? There's no echo or anything. Everybody can hear me okay? Great. All right. So let's get started. If you guys are new to it, just how I do it is I'm just going to ask you to keep your mics on mute. If you have any questions, I will stop towards the end of the section and ask. If you have any questions while you're going along, you can use the chat box. I'm here with David. He's the standby TA, so he can also help you out. Okay. So we are on our 15-week MCAT course. This is just some information about the course. All of the lectures are uploaded. The PowerPoints are uploaded to a folder that we send. I send them in the GroupMe. There's the link over there. The Instagram handle for our page is at Socratic Med _. You can find our YouTube page over here. And we do 3 lectures a week. We do them on Thursday, Friday, and Saturday at 8:30 p.m. Eastern time. And on Sunday, we hold free office hours where you can just stop by and ask us any questions as well. Okay, so with that, let's get right into it. So this is pretty much our learning objectives for today. We're going to be talking about the renal system, which obviously centrally involves the kidneys. So just to review some functions that the kidneys oversee, it removes nitrogenous waste from the body, metabolic waste from your cells. It helps balance water and ion levels in the body. It helps regulate blood pressure. It oversees erythropoiesis, which is the scientific term for red blood cell production, and it helps regulate calcium and phosphorus levels in the blood. So I'm pretty much going to spend this lecture just going through each of those bullet points, and we're just going to go into detail specifically how the kidney accomplishes all of this. Okay, so our first section is the kidneys and the urinary system. So the urinary system is also known as the excretory system. The kidneys, as you can see over here in this diagram to the right, there are these 2 sort of oblong organs on either side of the body. They're responsible for the removal of metabolic waste. In humans, that comes in the form of urea. That's the nitrogenous waste I was talking about before. And it can also remove bicarbonate, sodium, and water as well, among a couple of other smaller molecules. So blood enters the kidney through the renal artery because, as we know, arteries carry oxygenated blood away from the heart and to the target tissue. So the renal artery's target tissue is the kidney. It brings it in, and then purified blood leaves the kidney and re-enters the body, the systemic circulation through the renal vein. So waste products, ions, and water are absorbed by the kidney and are directed to the ureters. So you can see the kidneys over here. And then these 2 sort of yellow tan tubes that are coming off of each of them leading to the bladder are called the ureters. So we have 2 of them, and both of the kidneys empty into one bladder. So the ureter descends from each kidney and empties into the bladder. And when the bladder fills, it's lined with these receptors called stretch receptors, and they send signals to the pons, which is a part of the brain that we've discussed before in our previous lectures. The pons has what we refer to as the urination center. And when it receives that message from the bladder that it's full, it communicates with the cortex a little bit farther up in the cerebrum. So if the context is appropriate, if we're in the appropriate context to urinate, the parasympathetic nervous system allows for a contraction of smooth muscle around the bladder and a relaxation of the internal sphincter. So it's sort of like a simultaneous thing. When the bladder fills up, the smooth muscle is involuntary. Remember, it's dominated by the it's dominated by the autonomic nervous system. and specifically the parasympathetic section of the nervous system. So it contracts the smooth muscle around the bladder, which expels, it pushes the urine downward, and then the internal sphincter is one of two sphincters that we have. The internal sphincter is also involuntarily controlled, so that's also mediated by the parasympathetic nervous system. The external sphincters, actually, we do have motor control of it because it's striated muscle, so it's voluntary. So that's the sphincter that we control to release urine. Urine flows past the internal and then the external sphincter, and then out the body through the urethra. So that was sort of like a macro view of the urinary system. So we're just going to review this really quickly, and then we're going to start going deeper in. We're going to do the kidney, and then we're going to do the nephron, and then we're going to explore some other functions of the kidney. So let's start with this first question. Does anybody have any questions about what we just reviewed before we start? You can type in the chat. You can unmute really quickly if you need clarification for anything. Okay, great. So then I'm going to start the poll. Read the question. There are 4 answers. I'll give you guys about 1 to 2 minutes, and we'll regroup and discuss. And polls are open. All right, take about 15 more seconds. Remember, as David said before, the polls are anonymous, so just give it your best shot. All right, so I'm going to end the poll. Nice. Okay, so 22% of you said A, 67% said B, 11% said C. So the correct answer is B, mechanoreceptors. So we can go through the different receptors. Really, the only way you're going to get this question right is if you have knowledge about the different types of receptors and where they're found. So primarily through the use of A, chemoreceptors is not correct for a couple of reasons. There aren't chemoreceptors in the bladder, and that's not the way the bladder measures how full it is. Chemoreceptors are found in blood vessels, probably a couple of other parts of the body, but they help detect different chemicals in the body, like elevated levels of CO2, for example. They'll increase ventilation rate. C, photoreceptors are not true either. Those are found in the eyes. Those are your rods and cones. Any sort of receptor that is sensitive to light or electromagnetic waves are photoreceptors, so those aren't in the bladder. D, hygroreceptors. Nobody picked that, which is great because humans don't even have them. I just recently learned about them. They're found in certain insects, and they detect changes in atmospheric moisture. So B is the correct answer. It's mechanoreceptors, just because those stretch receptors that we talked about on the previous slide they're classified as mechanoreceptors because it's mechanical input that they're taking. They take the stretch. When it stretches too much, they communicate with the pons, and then micturition, which is the other word for urination, will either occur or not occur depending on the context. You guys seemed okay with that one. Does anyone have any questions before I move on? All right. So we're going to zoom in on the kidney a little bit. I put another diagram over here to the left. So once the blood makes it to the kidney, it's carried to the kidneys via the renal artery, as we were talking about before. And the outer region of the kidneys is known as the cortex. So a lot of structures you'll see cortex and medulla. Cortex usually refers to the outside of it, and medulla refers to the inside. So it's kind of just like the brain. I mean, we have a cortex and we have a medulla in there. So just think about that if you ever get confused which is which. The outer region is the cortex. The cortex is the outer region of the kidney, which has an ion concentration isotonic to that of plasma. So the cortex tonicity, the solute concentration, is basically the same as plasma. And we're going to talk a little bit later about why that's important, but I'm sure if you're familiar with osmosis, then you can already pretty much, understand why. So in the cortex, you will find the glomerulus and the convoluted tubules, which are two different parts of the nephron. They are toward the beginning when the filtrate just enters the lumen. So the inner regions of the kidney, which are right over here, these darker, like sort of pinkish areas, that's known as the medulla. That's the innermost part. It's the striated inside of the kidney, and it has a hypertonic concentration compared to its surroundings. So the medulla is very salty. It has a very high solute concentration, and that later we'll see that it helps pull water out of the nephron. So the difference in tonicity between the cortex and the medulla is essential for proper kidney functioning, like I just said. And urine leaves the nephron after it goes through everything, which we'll talk about in just a minute. It leaves the nephron and passes through the renal papilla into the calyx. So that's right over here. If you take a look at one of these triangles of the medulla, the renal papilla is that connection right between there and the calyx. The calyx is just this opening that empties into the renal pelvis. So there you could see that there are multiple medulla throughout the entire thing. They're constantly filtering your blood. They're making filtrate, and it's pouring separately. Each medulla is pouring separately into the renal pelvis, and then the renal pelvis makes its way out of the kidney and into the ureter. So someone asked me how many questions about the types of receptors are on the MCAT. You might not see too many that are specifically asking you about the receptors, but they pop up. You should know, the difference between mechanoreceptors versus chemoreceptors versus nociceptors. So they're not going to explicitly ask you. I wouldn't say there probably will be no questions on the MCAT that are just specifically asking you to identify them. But sometimes in the context of your reading, or if you're answering a question about something else, you will need that knowledge in order to answer it. So I would definitely know it. There aren't too many. There aren't too many to know, so it's not too hard to spend too much time. I think it's worth knowing. And when you say convoluted tubules, are you talking about proximal and distal ones? Yes, I am, and we're going to go over that right now. I believe all of that is on the next slide. Okay, so the next two slides, we're going to be taking a close look at the nephron, because this is a topic that the MCAT does like to test. So blood arrives from the body via the afferent arteriole and enters a bundle of capillaries known as the glomerulus. So over here, I underline the A in arrives and the A in afferent. I think that's a pretty helpful mnemonic for you to remember the difference between afferent and efferent arterioles. If you were studying, other systems like the nervous system I did a lecture on, we were talking about afferent and efferent neurons. So it's the same thing. We use the words to connote the same exact thing. Afferent is arriving, efferent is exiting. So in this case, the afferent arteriole connects to a bed of capillaries in the glomerulus. It goes into the glomerulus through those capillaries, and then it comes out the efferent arteriole and travels down the medulla and back up via the vasa recta. So the vasa recta are these sort of capillaries that are following this loop over here, which we'll later talk about, called the loop of Henle. So the pressure in the glomerulus and the glomerular filtration rate, which normal filtration is about 90 to 120 milliliters in humans, is regulated by the constriction and dilation of the afferent and efferent arterioles. So the glomerulus, just like any other capillary bed, is very sensitive and they're very fragile. In order for capillaries to do what they're meant to do, which is exchange nutrients and gas, they have to be super thin. So capillaries, actually, like some of the finest capillaries, blood cells will pass through single file, increases surface area, so there's gas exchange that's allowed. So they're very fine and they're very fragile. So we have to have very fine-tuned ways to control the blood pressure going in and out of the glomerulus. So one of the ways they do that is control of the afferent and efferent arterioles. So vasoconstriction of the efferent arteriole causes an increase in the glomerular filtration rate. So if you think about that logically, the efferent arteriole is the one that is leaving the glomerulus. So if you constrict that, you're stopping the flow of blood out of the glomerulus, but the flow of the blood in is remaining the same. So that's going to increase the blood volume in the glomerulus, and it's also going to increase the hydrostatic pressure. Likewise, the vasoconstriction of the afferent arteriole causes a decrease in glomerular filtration rate because it's cutting off the blood supply to the glomerulus. So if we cut off the blood supply by constricting the afferent arteriole, and we keep the efferent arteriole open, the glomerulus is going to lose blood volume and it's going to lose blood pressure, and the GFR is going to go down. And the same applies to vice versa. Vasoconstriction of the EA causes increase, vasodilation of the EA causes decrease. Filtrate from the blood is emptied from the glomerulus into Bowman's capsule and heads toward the proximal convoluted tubule. So Bowman's capsule is just that surrounding area of the glomerulus. The filtrate leaks out of the glomerulus and is collected by the Bowman's capsule, which is also known as the glomerular capsule. And it is like funneled in right over here and heads down to the proximal convoluted tubule. There's a lot of information on this slide. Does anybody have any questions about it? Specifically about pressure in the glomerulus and how pressure affects the GFR? Okay. We're going to continue a little bit with the nephron on the next slide. So if you guys are okay with this, then we'll move on. So the nephron continued. Filtrate, as I mentioned before, is the fluid that leaks into Bowman's capsule and goes through the entire lumen, this whole loop. It's a fluid consisting mostly of sugars, amino acids, urea, and it enters the proximal convoluted tubule from Bowman's capsule. So picking up where we left off on the last slide, the proximal convoluted tubule is responsible for about 70% of all fluid reabsorption. So sugars and sodium are actively transported back into the surrounding blood vessels, and water passively follows. So it's important to have an understanding of osmosis and how it works. It's not too hard conceptually in terms of the kidney. All you really have to remember is that water pretty much follows where the solute goes. So sugar and sodium gets pulled back up, water is going to follow. The filtrate then enters the descending limb and the loop of Henle. So the descending limb is highly permeable to water. As it descends farther into the medulla, the concentration of the surrounding tissue increases in ion concentration and drives water out of the filtrate. So when we're talking about the descending limb, we're actually talking about this on this diagram over here, this left side. So this top, like we mentioned before, the glomerulus and these top parts of the tubules are in the cortex. So those are isotonic, remember? It's basically the same concentration as blood plasma. So there's not going to be much fluid exchange there. Once you start, there's not going to be much fluid exchange passively. We have active transport for ions. So anything that requires ATP can go against its gradient. But in terms of passive transport, as we get farther down this descending limb of the loop of Henle, we get into the salty medulla. So it's high solute, high concentration, and that actually forces water out. Okay, so the thick section of the ascending limb of the loop of Henle actively transports ions back into the medulla to keep its high ion concentration. So the difference in permeability between both limbs to keep the concentration of the medulla constant is known as a countercurrent multiplier. And that's a concept that you'll see pop up on the MCAT in a couple of different places: the countercurrent multiplier, countercurrent exchange. The red structure right over here is the glomerulus. That's just that red ball right in the middle. That's where the afferent arteriole connects to the glomerulus, and it filters the blood and creates the filtrate to go to the rest. the red descending structure. If you're talking about this, those are the vasa recta. So the vasa recta are basically blood vessels that travel along the loop of Henle. So this blue, this blue is the deoxygenated blood, and this red is the oxygenated blood. And these purple crossing over is just, the oxygenated blood crossing over, getting rid of the oxygen into the tissues, and then leaving the veins. So like I was saying, the countercurrent multiplier is a concept that's going to pop up pretty often. So in terms of the nephron, we're talking about countercurrent multiplier in that the ascending limb is highly the red is the capillary and the blue is the vein. So it's countercurrent multiplier, just because water flows out into the medulla, which sort of would lower the solute concentration. But when you loop around and you start going back to the ascending limb, it actually filters the ions back out into the medulla. So it restores the concentration. So it's countercurrent is basically two things that are going in different directions, but it increases the efficiency of the system. So it's just going down natural gradients, and because they're countercurrent, it's efficient. I hope I'm explaining that well enough. If you'd like me to explain it differently or explain it again, then just type in the chat and I will try to explain that again. So the difference, we did that. The filtrate then enters the distal convoluted tubule, where reabsorption of urea, sodium, and water is highly regulated. Secretion of drugs and toxins occur here also. So the distal convoluted tubule, like I said, reabsorption occurs. Reabsorption is when the filtrate, stuff from the filtrate, is being absorbed back up into the tissue. Conversely, secretion is when something comes from the tissue into the lumen. So when we talk about secretion in terms of like endocrine functioning, we're usually talking about releasing into the bloodstream for a purpose. But when we're talking about the excretory system, secretion is drugs and toxins and things that we don't want that are being put into the lumen to be excreted. Yeah, so the main concept of countercurrent multiplier. So if we follow this descending limb, as we get lower, the outside becomes more solute concentrated. So that water, the water that's in this limb, is going to flow out just by passive transport, because there's high solute in here. It's going to flow out, and that you would expect that to cause a decrease in the solute concentration. But as it comes over here, we're actually pumping ions back into the medulla. So it's restoring the concentration gradient that the water was pulled out of. So it's going in two different directions, and it's sort of restoring. It's sort of like a cycle. So it has two stops, and they're sort of feeding in and on and off each other. So water goes out on the left side of the diagram, and then as you go up the other one, the ions go out and they sort of restore. The purpose of countercurrent multiplier. So it's the purpose of the countercurrent multiplier is, it's to keep water flowing back in, and it's also to improve the efficiency, because if it weren't countercurrent, then we would have to use ATP a lot more often for active transport if we weren't sort of setting this up in a way that it would reestablish the gradient. When the body has excess water, we'll talk a little bit about blood volume and excess water, but basically when it has excess water, the distal convoluted tubule is not permeable to water, and it just gets excreted with urine. You have very dilute urine, and it exits that way. I don't have too many like mnemonics or ways to recall this information. I know a lot of these biology courses, it really is just straight information, so you're kind of just going to have to commit it to memory. Okay, so we said the filtrate enters the distal convoluted tubule, where reabsorption of urea and sodium occur. Secretion of drugs and toxins also occurs in the distal convoluted tubule, remember? And then the filtrate enters the collecting duct, which is basically the last stop of the nephron. and that's where the secretion of potassium and hydrogen ions occur. So we'll talk a little bit later also about the regulation of hydrogen secretion and also about water flow in and out of the collecting duct. So the duct empties out of the nephron and continues out of the kidney to the bladder. So like we were just talking about before on the other slide, after it leaves that collecting duct, it comes out of the calyx and goes into the renal pelvis and out the ureter. Okay, so we're going to do one more review question before we go on, and I'll give you guys about a minute, and then we're going to discuss. So take a minute look. If you have any questions, again, you can type in the chat. We'll take this time to answer some of those. Okay, so the poll is live. Give it your best shot. All right, give you guys about 10 more seconds, and then we're going to close the poll and go over it. Okay, so I'm going to close the poll. And I will share the results. So it looks like the most popular answer was B, but there was a decent spread. So 27% said A, 45% said B, no one said C, and 27% said D. So the correct answer is actually B. So good job to those of you that did get it correct. So we're going to assume an individual's glomerular filtration rate drops to 60 milliliters per minute. So if you remember before, 90 to 120 milliliters per minute is actually the average GFR for a human. So dropping to 60 is pretty dangerous. Vasoconstriction of the afferent arteriole A would actually cause this to decrease even more. So remember, the afferent arteriole is arriving to the glomerulus. So if you cut off blood flow to the glomerulus, you're actually going to decrease the glomerular filtration rate just because not as much blood per minute is getting to the glomerulus. C, decrease in ADH production. We're going to talk in a couple of minutes about ADH. It's antidiuretic hormone. If you know what it was, you know that it works to increase blood pressure, so it would increase GFR. Even if you didn't know what it was, you could still answer the question just by knowing that B is correct, but we'll come back to B in 1 second. Decreasing the permeability of ions in the collecting duct. That would also decrease because, again, what we'll talk about in a little bit is aldosterone actually increases permeability and causes water to come back in to follow the ions up through the collecting duct. But just based on what we talked about before, you should know that vasoconstriction of the efferent arteriole actually causes an increase in pressure in the glomerulus. So an increase in hydrostatic pressure is going to bring the glomerular filtration rate up. So out of all 4 of those, the most likely response from the kidney in order to return GFR to normal would be vasoconstriction of the efferent arteriole. Is this the one you were talking about? Okay, so the afferent arteriole you can see right here is coming in. It's from this artery. So if you see this artery that's going all down here, that's coming from systemic circulation. So the blood is following up the afferent arteriole because it's arriving at the glomerulus. And then the efferent arteriole, it's efferent, but it's still an arteriole. So it's still carrying oxygenated blood. And this says to systemic circulation. So eventually it's going to empty into systemic circulation, but it goes through the nephron first. So it goes over here, and then these capillaries are known, and blood vessels are known as the vasa recta. So it goes down here and then crosses over, drops off its oxygen, picks up any waste products, and then it leaves through the renal vein. So filtration rate just means the blood that enters the glomerulus. Vasa recta is over here, by the way. It's on the slide right over here, that second bullet point. Yeah, so glomerular filtration rate is the amount of blood that passes through the glomerulus that is filtered per minute. So about 90 to 120 milliliters of your blood is filtered in the glomeruli of your body per minute. Okay. Okay. So then, yes, pressure does cause the GFR to go up. Any increase in pressure is going to cause GFR to go up. So let's go back over here. Just because it's the hydrostatic pressure. So if we're increasing pressure in the capillary bed, there's more outward pushing of the solutes and everything that we need to get through the capillaries, because the endothelium of the capillaries leak and they can expand and they can allow things to move through them. That's just a property of capillaries. That's how they work. So if you increase the pressure systemically, it's going to follow through the glomerulus and it's going to increase there as well. It's going to increase the glomerular filtration rate. That's actually why high blood pressure is so dangerous, because these organs that have such delicate capillary beds, if your blood pressure is too high, it's going to destroy the capillaries, and then it's going to lead to tissue failure, and then eventually it could lead to organ failure as well. So constricting the efferent arteriole, remember the efferent arteriole is leaving, so it's after the blood passes through the glomerulus. So if you constrict that, you're cutting off the amount of blood that can flow through it in a certain amount of time. So if you're sort of plugging it up, and the blood is still coming in from your body, but we're plugging up that efferent arteriole, so there's going to be more and more blood that's entering the glomerulus than is actually leaving. So that's how we're increasing pressure in the glomerulus, and the GFR increases as well. That's an important concept, how constricting the efferent arteriole and the afferent arteriole as well too. So the afferent one is before it. So if we constrict the afferent arteriole, no blood is going to make it to the glomerulus. So that's why it decreases GFR in the glomerulus. Cool. You guys have great questions tonight. Okay. So we're going to move on. We're going to talk a little bit about the other functions of the kidney too. Okay, so these are two that we're going to talk about. You absolutely do not have to know what they look like. I just thought it was cool. It was a cool visual for vasopressin, AVP, and aldosterone. So the first one we're going to talk about is ADH. Also, like on the other one, it's also known as vasopressin. This one on the left is ADH, antidiuretic hormone. It just has a bunch of different names. Arginine vasopressin, argipressin, AVP, ADH, vasopressin alone. So it's a hormone that is released by the posterior pituitary in response to an increase in blood osmolarity and a decrease in blood volume. Pretty self-explanatory. When the solute concentration in the blood is too high, there are osmoreceptors in the hypothalamus that fire and stimulate the pituitary to release antidiuretic hormone. So the hypothalamus is the solute control center of the body, and that's pretty much what mediates the release of ADH. So ADH next acts, on the distal nephron, mostly on the collecting duct, like we were talking about a little bit before, to increase its permeability to water. So the collecting duct without the action of ADH is not really permeable to water. So if we don't have any ADH production, water is going to pass through the collecting duct, and it's really not going to be absorbed. It's going to pass on down the ureters, down into the bladder, and it's going to be excreted as urine. So when we add ADH, antidiuretic hormones, it acts on the distal nephron, and it inserts these little channels called aquaporins. So aquaporins allow the collecting duct to be permeable to water, and water flows passively out of the collecting duct into the surrounding tissue. So yeah, the cells lining the collecting duct produce tiny channels, so they sort of make it, they have the genes for this channel, and then ADH turns them on. So they start expressing these aquaporins, and the more aquaporins that are expressed, obviously, the more permeable to water the collecting duct becomes. And a fun fact, alcohol inhibits the release of ADH. So how do we experience this? If you drink a lot of alcohol, you may have noticed that you have to pee a lot. And that's because it's inhibiting the release of antidiuretic hormone. So all of this water is just leaving your body. You're not able to absorb it. And that's also why alcohol can cause dehydration. Yep, same with coffee. So aldosterone, while ADH responds to high solute concentration in the blood, aldosterone is secreted usually when blood pressure is detected to be too low. So the way we do that, aldosterone is secreted by the adrenal cortex when blood and osmolarity or blood volume is too low. So the adrenal cortex, let me see if I have a picture of it. If you look right over here at these two kidneys, these two triangles sort of looking organs that are sitting on top, those are the adrenal glands. So adrenal, the word AD means on top. Renal means kidney. So it's literally the kidney, the organ sitting on top of the kidney. And we'll definitely talk about the adrenal gland in the endocrine lecture. But for this lecture, I just want to keep it in terms of what we're doing here, in terms of the kidney. Okay. So like I said, yeah, when blood osmolarity or blood volume is too low, aldosterone can be secreted. It's also a part of the Renin-Angiotensin System, which we'll talk about in another couple of slides, which is another really pretty high yield thing on the MCAT. The hormone also acts on the distal nephron, just like a lot of the other hormones. But aldosterone, instead of increasing water uptake like antidiuretic hormone does, aldosterone increases sodium reabsorption in the distal convoluted tubule and the collecting duct. So aldosterone and ADH work together to raise the blood pressure via ion concentration. So it's sort of like a 1-2 punch. Aldosterone is going to increase the solute concentration in the blood. And like we were talking about before, wherever solute concentration increases, water is going to follow. But water can't follow unless it can pass through these barriers. So the aquaporins that are put in by ADH allows water to be reabsorbed back into systemic circulation. So again, just that key concept coming up that wherever the solute goes, the water follows. And both ADH and aldosterone work to increase blood pressure. So you can keep that also as a quick note. So increase in aldosterone, increase in ADH usually signifies an increase in systemic blood pressure. Okay, and we're going to talk really quickly about regulation of pH. It's not super high, but I think it's definitely worth mentioning. So the kidneys are also essential in maintaining blood pH. They're not the primary method for which we do maintain blood pH, but it's definitely essential. So physiological pH is about 7.4. Carbonic anhydrase, you don't have to know too much about the enzyme. You just have to know that it is found in the kidney and it helps convert carbon dioxide into carbonic acid. So it's found specifically in the thin parts of the loop of Henle. Yeah, and it creates carbonic acid. So that carbonic acid quickly breaks down into bicarbonate ions and protons. So the kidney can actually use this breakdown of carbonic acid to pick and choose the pH. So, bicarbonate ions, wherever they are, they're going to increase the pH because they're basic, and protons are acidic. The very definition of an acid is just the proton concentration, how acidic it is. So the kidney can basically pick and choose what it wants to excrete and reabsorb, and it can affect blood pH that way. So it can reabsorb or secrete bicarbonate ions and protons as needed. So if we have, if our pH is dropping pretty dangerously low, 7.1, 7.0, we can excrete protons in urine. If it's 7.1, 7.2, we can excrete protons. That's usually the case. It's usually the case that it's some sort of acidosis and that protons have to be excreted and bicarbonate ions have to be taken up. But if by some chance the body is too alkaline, 7.6, 7.7, then the opposite can also occur. We can keep protons and we can expel bicarbonate ions. But like I said, it is a very long process and it can take days for blood pH to return to normal, even just from a couple of decimal points. Return to the optimal level, just because, you can only excrete protons so fast. But the respiratory adjustments to pH, like hyperventilation, they work very quickly. It takes usually minutes. So hyperventilation, as if you do something like increase your physical activity, it's going to result in excess CO2, excess lactic acid in your blood, and it's going to cause a decrease in blood pH. So the chemoreceptors in your blood actually communicate with your brain and your lungs, and it increases the respiratory rate. You hyperventilate, you get rid of the CO2, and it brings your pH back up to 7.4 relatively quickly when compared to how the kidney does things. So ventilation, respiratory adjustments to pH is the primary way that we balance our pH, but the kidneys are an essential part, so it's definitely worth mentioning and definitely worth knowing. Okay, does anybody have any quick questions before we go on to the next review? Okay, so I'm going to relaunch the poll. I'll give you guys a few minutes to read this and answer. Okay, so polls are open. Again, these are anonymous, so try your best. All right, take another 10 or 15 seconds and I'm going to close the polls. Okay. All right, so 27% of you said A, 36% of you said B, 27% of you said C, nobody said D, and 9% said I have no idea. So the correct answer is in fact B. So let's go over it. A patient is entering diabetic ketoacidosis, a condition in which excess ketone bodies are synthesized due to increased fat metabolism. How might the body react to this situation? So ketone bodies, as you know, some of the most common ketone bodies are acidic. So when someone enters diabetic ketoacidosis, as you may be able to decipher from the name if you're not familiar specifically with the condition, it causes your body to drop pH, and it can be very dangerous for metabolic functioning. And that's due to the fact that insulin is not being produced, or insulin is not able to be utilized by the cell, so it can't take up any glucose in the blood. So normally glucose metabolism is one of our main sources of energy, but because we can't use it, we switch over to fat metabolism. Diabetics switch over to fat metabolism, and the excess of ketone bodies can cause ketoacidosis. So a decrease in ventilation rate, this would not help the situation. This would make it worse. Diabetic ketoacidosis often causes hyperventilation because, like we spoke about before, that's the way that your body really restores pH the quickest. So increased proton secretion, B is the correct answer, because as well, like we talked on the slide before, this proton secretion would mean that protons are being emptied into the collecting duct and into the distal convoluted tubule, and they're being sent out of the body. So remember, secretion is when the surrounding tissue puts stuff in order to get rid of it from the body. So we're secreting protons, which means we're getting rid of what's making the body so acidic. So a decrease in ADH production would stop water from coming into the body. I don't know if it would have a direct effect on diabetic ketoacidosis. It may, but if you're just following the logic, if you're decreasing ADH, then you're not letting water into the body, which can only make the environment more acidic. You're not diluting the body, you're not letting water back into it. So C can't be the correct answer either. And D, for the same logic that C is, if we allow ions into the body, then water is going to follow. But we're not doing that. We're decreasing aldosterone production, so it's not helping get the body back to its natural equilibrium, that's 7.4 physiological pH. The only one that definite concrete lowers pH levels is increasing proton secretion in the nephron. So in regards to choice A, is it that proton ion concentration would increase because the body would break down CO2 to bicarbonate and H thereby increase H in the body? Yes, so diabetic ketoacidosis is these ketone bodies, they're acidic. So when you're acidic, you're giving off these H+, these protons, and that's what lowers the pH. So if we decrease the ventilation rate, we're not getting those out of our body quickly enough. Does that make sense? And it's important that you understand, for B to be correct, it's important you understand what secretion means. So proton concentration is increasing in the body as we become more acidic, yes. So in order to get it back down, we have to secrete those protons. We have to get rid of them. Secretion basically just means getting rid of them. So if we're getting rid of the protons, we're bringing it back up. CO2 is acidic. You can consider CO2 itself is not considered. It's not acidic, but the way it breaks down, it combines with hydrogen, it forms carbonic acid, so it is acidic. And that's actually, during hyperventilation, we're releasing CO2, so we're actually making our body more basic. So if we're below physiological pH, 7.1, 7.2, by releasing CO2, we're actually bringing the pH back up to 7.4. And that's why we hyperventilate. So people in diabetic ketoacidosis will actually experience the need to hyperventilate because it's their body's mechanism. It's these like homeostatic compensatory mechanisms that try to bring you back up to optimal pH. And yep, that's an interesting little tidbit about DKA also. The 3 Ps, polydipsia, polyuria, and polyphagia. So thirst, a lot of pee, and a lot of appetite. That's a pretty good thing to know too, actually. Yes, CO2 can also produce bicarbonate ions, but we don't breathe out bicarbonate. So what we do breathe out is more acidic, and that's why we get back up to that pH. It does produce the bicarbonate ions, so in the kidneys, we have the choice. We have hydrogen ions and bicarbonate ions, so we can choose which one we're expelling. So if we're too basic, we can get rid of the bicarbonate ions, and we're going to bring our pH down. If we're too acidic, we can get rid of those hydrogen ions, and it'll bring us back up. So the fact that CO2 produces bicarbonate ions and hydrogen ions is actually helpful in that case. So yes, good point. Okay. All right, so now we're going to talk about the Renin-Angiotensin System. So you can look this up. It has a couple of names, the Renin-Angiotensin Aldosterone System, as it's shown right above the diagram. I was trying to pick a diagram that was easy to read, simple enough that it gives us the bare bones of exactly what we need for the MCAT. So I think that this is a pretty good diagram. So you should definitely be familiar. You should definitely know everything up until these last things down here, and you should definitely at least be familiar with the trend of what these are doing. So okay, these are a little bit out of order. So I'm gonna do this one first, and then we're gonna hop back to the other one, just because we're already talking about the renin-angiotensin system. So you know what, we're gonna do this first. So the juxtaglomerular apparatus and the macula densa, these are two concepts that we should probably know before we discuss the renin-angiotensin system. So we will do that. That's why I did that. The afferent arteriole maintains direct contact with the distal tubule. So if you look right over here, we have the afferent arteriole over here on the bottom, this red vessel on the bottom. And then the distal tubule is right over here. Okay, so the cells lining the afferent arteriole at this point are called the juxtaglomerular cells. So you can see these little red cells lining like these little epithelial cells. The green mesh, the extraglomerular mesangial cells, I wouldn't pay attention to those. In my studies and in my MCAT experiences, I've never come across anything with them. So I think it's probably just out of the scope of the MCAT. So don't get too caught up over there. But right now we're looking at the juxtaglomerular cells, which are lining this afferent arteriole. So these are baroreceptors, which means they can respond to changes in pressure in your blood vessels. So they specifically respond to low systemic blood pressure, and they actually secrete renin, which, like I said, we're going to talk about on the next slide. So these cells are touching up against the distal tubule, like I talked about, and the cells that are lining this sort of cross-sectional area at the exact point that it touches the afferent arteriole, these cells are known as the macula densa. So the macula densa are cells that are chemoreceptors, unlike the ones that are lining the afferent arteriole cells. The JG cells are baroreceptors. The macula densa are chemoreceptors. So these detect osmolarity in the filtrate. Specifically, when the osmolarity is low, the cells increase filtration in two ways. So the first is that they stimulate the JG cells to release renin, as we mentioned above, and we're going to talk about in 15 seconds. And two, they directly dilate the afferent arteriole to increase blood going to the glomerulus. So we talked about it a couple of times on a couple of other slides, and it's popping up again. So remember, when you dilate the afferent arteriole, we're making it bigger. The afferent is what's coming in. So blood is coming from the body, from the heart up into this afferent arteriole, into the glomerulus. So if we expand this afferent arteriole, we're letting more blood into the glomerulus at a time. So it's going to increase that filtration rate. And now for the main event, the renin-angiotensin aldosterone system. The JG cells surrounding the afferent arteriole secrete renin when there is a drop in systemic blood pressure. So renin is just that chemical. The substrate for renin is angiotensinogen. So renin is the enzyme that attaches to angiotensinogen, which is a precursor hormone that is produced by the liver. So it's constitutively produced by the liver, and it's basically just chilling in your body, circulating, waiting for renin. And the reason it does this is because the RAAS system can act pretty quickly. And it can act pretty quickly because we already have the supply of angiotensinogen. So instead of waiting for the liver or some other organ to create the final form of angiotensin, we create a precursor and we just let it float around. And then when we're ready to activate it, we secrete renin, which starts the cascade to activate it. So like we just said, the first step is renin turning angiotensinogen into angiotensin 1. And you can see right over here, it displays that pretty clearly. We have angiotensinogen, then it comes from the kidney, renin, it interacts, and then we have angiotensin 1, which is still a precursor hormone. It's not active yet. The lungs then, interestingly enough, produce angiotensin converting enzyme, which is called ACE. So I'm sure if you're familiar with medicine, or you're familiar with anybody that is over the age of 50, you've heard of ACE inhibitors, and those control blood pressure. And this is exactly why. We can see why. So the lungs produce ACE, angiotensin converting enzyme, which complexes with the first form of angiotensin to create angiotensin 2. So angiotensin 2 is the complete form. It's the final form, and it's active. So that systemically raises blood pressure. And it increases blood pressure by everything over here at the bottom. So the kidneys, it constricts glomerular efferent arteriole and increases NAH exchanger activity. It acts on the posterior pituitary to increase ADH secretion, like we talked about before. That's going to allow us to bring water into the body, and it's going to increase the blood volume, which increases hydrostatic pressure. It acts on the vascular smooth muscle cells, which line your blood vessels, and that creates hypertension. That's just vasoconstriction, your basic vasoconstriction throughout the body. The hypothalamus, it stimulates thirst in the hypothalamus, which allows us to intake more water, again, increasing blood volume and increasing hydrostatic pressure. And it works on the adrenal cortex to secrete aldosterone, like we mentioned before, which does the same thing, increases solute uptake and increases water flow back into the body. And that acts pretty quickly, so it's a pretty acute system. Does anybody have any questions about how that works? I don't really think you will need to know much more for the MCAT than what's on this slide. The concepts that they really like you to understand is that angiotensinogen is always there, and it's regulated. This whole system is not regulated by angiotensinogen, but it's started by renin. And then that ACE comes from the lungs. They like to talk about ACE inhibitors, and they like to talk about how they affect the body. So ACE, we're always increasing blood pressure, systemic blood pressure. ACE inhibitors are always going to bring it down because we're going to stop all of this stuff from happening. Anything downstream from ACE, if you use an ACE inhibitor, it decreases it. Okay, does anybody have any questions before we move on? Lovely. So we're going to do another review question, and this one involves ACE inhibitors. So ACE inhibitors are widely used treatments for hypertension. For whom would it be dangerous to take an ACE inhibitor? So think a little bit about that. We're going to regroup in a minute or two, and we'll discuss the answers. Again, these are anonymous, so try your best. I will start the polls right now. Polls are open. Take about 15 more seconds. Okay, I'm going to end the polls. Nice. Okay, so 13% of you said A, nobody said B, 25% of you said B, 50% of you said D, and 13% of you said I have no idea. So the correct answer is in fact D. ACE inhibitors are widely used treatments for hypertension. For whom would it be dangerous to take an ACE inhibitor? So we know what ACE inhibitors do. ACE stands for angiotensin converting enzyme. So what it does, an ACE inhibitor inhibits that, so it's lowering our blood pressure. So you have to think, why would it be dangerous for someone to take a medication that lowers their blood pressure? And the most common answer would probably be because their blood pressure is already too low, or because something that blood pressure affects is already too low. So a person experiencing much higher than usual solute levels in the blood would not necessarily be dangerous for someone to take an ACE inhibitor, because high solute levels are actually a cause of hypertension. If people have hypertension, their doctor often tells them to stay away from salty foods because there's an excess of solute, usually sodium or chloride, that is in the blood, and that's what's actually retaining water and increasing hydrostatic pressure and increasing hypertension. So A would probably be a pretty good candidate for an ACE inhibitor. B, a person with chronically elevated ADH levels. Similar to A, if you have chronically elevated ADH levels, you're going to be having more water in your blood vessels. So again, that increase in that hydrostatic pressure, we're going to be hypertensive. ACE inhibitors would probably help, not hurt. A person with chronically dilated afferent arterioles, when the afferent arterioles are dilated, that means the glomerular filtrate rate is being increased because we're increasing pressure inside the glomerulus. But if we're chronically dilated, it's probably not a good thing because these capillaries are so sensitive and they're so fragile. An ACE inhibitor to lower that pressure would probably not be a bad thing. The only one that's pretty clear cut why it should not, the person who should not take an ACE inhibitor would be someone with an unusually low blood volume. So say they lost blood for some reason, or they don't have enough water in their blood, they would be considered hypotensive because their blood pressure is below the acceptable amount. So if you gave a person like that an ACE inhibitor, then their blood pressure is going to drop even lower, and then they'll probably enter shock or some sort of state like that, and it could be potentially fatal. Does anybody have any questions about that question?
Yes, Chris, I have a question.
Sure.
Chris, can you hear me?
Yes, I can.
Yes, I thought volume and pressure is inversely related, so that's why I didn't think that giving them ACE inhibitor, if their volume is low, that means their pressure is high, right?
No, so low blood volume is low blood pressure. So when our blood pressure is low, a lot of times the first thing that a doctor will look to do is to get fluids in you. So they'll ask you to drink water, or they'll hook you up to an IV drip, because low blood volume actually decreases the hydrostatic pressure inside your blood vessels. So there's not enough blood volume. Think about it. There's not enough fluid that's pushing out on your blood vessels. So the pressure lowers. The reason why, say, someone that has such a high sodium intake, like, the average American diet, we consume so much sodium. So the reason why hypertension is such a problem in the country is because there's too much sodium in the body. So it's keeping all of that water in. It's keeping this high blood volume, which is causing hypertension.
Why did you say dilated afferent arterioles? What's the deal with that? I thought the question before, we were constricting them to raise our blood pressure.
So when you constrict efferent arterioles, it raises the pressure in the glomerulus.
Oh, efferent, not afferent. Okay.
So when you dilate afferent arterioles, the person with C doesn't necessarily have high blood pressure, like systemically, but when you have dilated afferent arterioles, you're going to have high blood pressure in that glomerulus, which is a problem if it's too high for too long, because they're so sensitive. So ACE inhibitors will lower the pressure going into the afferent arterioles and lower the pressure going into the glomerulus. So it wouldn't be dangerous. Does that make sense?
Okay.
Sure. All right, so we're also just going to do a quick review. We're almost done with the lecture. We just have like one more little section, just a quick review of the other endocrine functions of the kidney. The stuff that we just went over is mostly the highest yield when it comes to the renal system, but the kidneys are pretty diverse and they have some other functions that I think are worth mentioning. So one hormone in your body that's part of another system that acts on the kidneys is calcitonin. Calcitonin is a hormone that is secreted by the thyroid that helps lower blood calcium levels by multiple mechanisms, the scope of which is outside of this lecture. But what you do have to know is that it's secreted by the C cells in the thyroid when blood calcium levels are too high. So there are specialized cells. We know that the thyroid controls metabolism as well, but there are specialized cells known as C cells in the thyroid. And when blood calcium levels are too high, these C cells, they secrete calcitonin. So there are a couple of ways that calcitonin works. You can look to the diagram to the right. Calcium deposition onto the bone, calcium resorption back into the blood. It acts on the small intestines too, but in the scope of this lecture, all I really care about is the kidney and how that works. And it's pretty simple as well, the concept. It just acts on the tubules to inhibit reabsorption of calcium ions. So basically, your nephron would absorb calcium ions normally, and if there's too much in the blood, calcitonin is released, and it tells the kidney to excrete them instead of absorb them back up. That's really all calcitonin has, all the effects calcitonin has on the kidney. Okay. So on the other side of the coin, parathyroid hormone is sort of like an antagonist calcitonin, or it sort of antagonizes calcitonin in that it has the opposite effect. So it's secreted by the parathyroid gland, and its effect is opposite that of calcitonin. So when blood calcium levels are too low, PTH, parathyroid hormone, is released. So through a number of mechanisms, PTH increases blood calcium levels. And again, we can look to the left if you want. Like I said, I'm going to post these slides, so you can always stop them, study them, and do some of your own independent research based on what you find over here. But it acts on bones. It releases calcium and phosphorus, reduces calcium and vitamin D activation in the kidneys, which is what we're really looking for. It's more so the calcium thing, the vitamin D. I'm not too familiar with it. You can definitely look it up on your own time, but really parathyroid hormone and calcitonin, it's really important to understand the ion flow in terms of both of them. And then it also, again, it has an effect on absorption in the intestines. So just to go back to the bullet points, in the kidneys, PTH has two effects. It blocks the reabsorption of phosphate into the proximal tubule, and it promotes the reabsorption of calcium in the ascending limb of the loop of Henle and the distal tubule and the collecting duct. So it's important to understand also why blocking the reabsorption of phosphate in the proximal tubule helps increase blood calcium levels. So calcium phosphate is the main form of solid calcium in the body. So when calcium and phosphate it precipitates out of solution and it becomes solid. So calcium phosphate is basically what your bones are made of. And in order to get rid of the calcium phosphate and keep the ion concentration high, we have to get rid of the phosphate. So we keep the phosphate out of the blood so it doesn't react with the calcium and take it out of the blood. So without phosphate, calcium is just a free ion, and that increases the calcium concentration. So that's what PTH does in terms of phosphate. And it also promotes the reabsorption just of the calcium ions in the ascending limb, puts them back in the blood. So it increases, it actually increases the calcium ion by taking calcium back up. And it also increases free calcium by blocking phosphate from coming into the system. Does anyone have any questions about that? Okay. All right, and another function of the kidneys worth mentioning is called erythropoiesis. So the kidneys are responsible for the synthesis of red blood cells. So that process is known as erythropoiesis because red blood cells are also known as erythrocytes. So in response to systemic hypoxic condition, hypoxic, hypo meaning low, oxic meaning oxygen, the kidneys synthesize erythropoietin. So this is a response to hypoxic conditions. If we have low hemoglobin levels, if we have low red blood cell count, and we're not getting enough oxygen to our cells, the response is to stimulate the secretion of erythropoietin from the kidneys. And erythropoietin is also sometimes shortened to EPO. So EPO travels to the bone marrow, where, if you remember, is the site of blood cell production, both red and white. It increases the differentiation from progenitor cells, those cells that are coming from stem cells to differentiate. And it basically just directs them to become red blood cells. So as oxygen levels return to normal, EPO levels drop. So it's sort of a pretty good example of a negative feedback loop, and it's a pretty easy one to understand. So oxygen levels go down, erythropoietin levels go up. When erythropoietin levels go up, oxygen levels go up, erythropoietin levels go back down. And it's sort of just like that. I mean, you can take a look over here. Erythropoietin promotes the production of mature red blood cells in the bone marrow. More red blood cells in the circulation lead to increased oxygenation and lower levels of hypoxia-inducible factor, suppressing EPO production. So that's the specific mechanism. You can memorize it if you want. I don't really think it's too high yield or too, too worth it. Okay, then we're coming to the bone marrow. Iron is necessary as well for red blood cell production. Its absorption and transport are also promoted by hypoxia-inducible factor, obviously because iron is a part of heme, and heme helps us carry oxygen throughout our body. And hypoxia-inducible factor, like we just mentioned up at the top, is degraded under conditions of normal oxygen tension. So in anemia or hypoxia, it promotes gene transcription of erythropoietin necessary for maturation of red blood cells. So basically exactly what we just said. They're just adding that extra step. So when hypoxic conditions are induced, this hormone known as hypoxia-inducible factor increases the transcription of erythropoietin from the kidney. Does anybody have any questions about that? I believe we have one more question. So this is the last question of the lecture. So take a look. A patient is experiencing shock, a condition in which blood pressure falls to dangerously low levels. What is the least likely response from the kidney? So take a look at this. I'll give you a minute or two, then we can discuss. And the poll is live. All right, I'll give you guys about 10 more seconds to get your final answers in. Okay, I'm closing the poll. Nice. Okay, so 20% of you said A, no one said B, no one said C, and 80% of you said D, and nobody said I have no idea. So yeah, the answer is actually D. That was a great job. You guys are doing really good at this stuff. So let's go over it. A patient is experiencing shock, a condition in which blood pressure falls to dangerously low levels. What is the least likely response from the kidney? So the first thing you have to look at, what is the least likely response? Make sure you're looking for something that the kidney would not do in low blood pressure situation. So an increase in erythropoietin secretion, it's not something, I wouldn't consider it the least likely response, because if we have low blood pressure, then we're not going to have good oxygen levels. Low blood pressure, excuse me, usually indicates hypoxia. So there's probably a pretty good chance that erythropoietin is going to be synthesized. And it's important to remember that that word secretion has different meanings when you use it in different different contexts. So if we're talking about the secretion of a hormone or the secretion of an enzyme, we're putting it into our system. So the kidneys secrete erythropoietin into the bloodstream. If we're talking about secretion in the urinary tract, then we're putting stuff into the filtrate to excrete it from our bodies. So it's the terminology is kind of confusing, but I put that answer in there specifically for that reason. So make sure you understand that the term secretion changes depending on the context. It's almost always going to be the same definition when we're talking about hormones, except when you're talking about secretion in terms of the urinary tract. So an increase in aldosterone production would increase blood pressure. So that is a likely response of the kidney. It's going to try to increase solute reuptake, and it's going to try to increase that water flow back in to increase the hydrostatic pressure and increase the systemic blood pressure. So B would be a likely response. Same thing with C, increasing the concentration of aquaporins in the distal nephron. That's just increasing ADH, antidiuretic hormone, and that follows with the increase of aldosterone. So if we let the water through, that's what we want. It's gonna let the blood pressure increase, and it's gonna save us. So the least likely response would be the constriction of the afferent arteriole. It is, this is not necessarily affecting systemic blood pressure, but we have low blood pressure systemically already. So our GFR, glomerular filtration rate, is going to be very low right now. So your kidneys acting, independently as a kidney, they're seeing that this GFR is low, and the kidney is going to try to increase it as much as possible. So it would actually dilate the afferent arteriole, and it would constrict the efferent arteriole because it wants to increase the pressure in the glomerulus. So constriction of the afferent arteriole would definitely be the least likely, because it's just going to drop the GFR even lower. And then the kidneys are virtually going to be filtering no blood per minute. You guys did a great job with that. Does anybody need any clarification on that? Okay, that's pretty much all of the content.
Hey, Chris.
Yes, hold on one second, Opal. I just have a quick question in the chat that I'm going to answer, and then we can do you as well. So quickly restating the ADH and aldosterone functions. Let me see if I can go back to them. So ADH is antidiuretic hormone. When solute concentration is too high, antidiuretic hormone is released, and what it does is it inserts these aquaporins in the collecting duct, which is the last part of the nephron. So at the last part of the nephron, when we secrete ADH, it allows water to come up. Because remember, the collecting duct is not permeable to water on its own. So ADH stimulates the production of these aquaporins, so it allows the permeability of water. It allows water to passively flow out of the filtrate and into the surrounding tissue. And then aldosterone is similar. It's a response to low blood pressure. Specifically, it's detected when blood pressure is too low, and then when blood osmolarity or blood volume are too low. So it just increases the blood osmolarity. It just allows us to uptake ion concentration. Again, in the distal convoluted tubule and the collecting ducts. No problem. Okay. Yeah, and then secretion, when we're talking about the urinary tract, the renal system, secretion is just putting it into the lumen, into the filtrate. The filtrate is what we're getting rid of from our body. So when you're talking about secretion in terms of the renal tract, we're putting it into the filtrate and we're getting rid of it from our body. When you're talking about secretion, basically with anything else, like endocrine functioning, when we're releasing, like when we're secreting epinephrine, for example, anything like that, secretion is releasing the product into our blood for systemic circulation. And another question? Opal?
Yes, can you go to the GFR slide, please? Because the questions I've done when it comes to GFR are way more complicated because now they're putting in inulin, which is this compound that just goes straight through the body without being filtered.
Wait,
How does that affect GFR? Or is this beyond the scope? Because I've done questions.
I'm sorry,
I
can't understand you very well, Opal. Can you mute one of your devices?
Okay.
I'm sorry. Can you hear me now? Yeah, I can hear you now much better.
Yes. So the question I've done with GFR,
yeah,
is the molecule called inulin, which goes through the body unfiltered. Explain how inulin affects GFR and why do they do these blood tests with inulin?
I'm not 100% sure, so I don't want to give you any inaccurate information, but I can definitely get back to you on the GroupMe. I haven't seen too much on inulin. I was looking, when I was making the lecture, I was considering whether to put in like inulin, creatinine functioning, and like clearance in terms of glomerular filtrate, because I'm sure you've seen that with your studies. I'm not too certain specifically the function of inulin, but I can definitely get back to you on it. I don't think it's too high yield, so I wouldn't worry too much about it, but I definitely would like to answer your question, so I'll get back to you after the lecture.
Sure, but shortly, if it's just passing through the system, it's not filtered, it's not going to affect GFR, probably.
Yeah, right.
Okay.
Okay. I will definitely get back to you on that. Okay, does anybody else have any other questions they want to go over? And if you think of anything else after it ends, you can definitely, join us tomorrow, 8:30. I am going to be on office hours with, I believe, Janelle tomorrow. So we'll answer your questions. If you have any questions you want us to take a look at in advance, you can definitely email us or you can send it in the GroupMe and we'll go over it as well. Hold on. Okay. I don't have a list of the highest yield content on the MCAT. At least I'm not familiar with, but that might not be a bad idea for us to look into actually. So I can definitely take a look into that. Most of the content that we do cover here, the course is structured around just the highest yield content. And yeah, I can definitely go over the countercurrent exchange. David actually typed out a much better explanation of it than I did. So I'm actually just going to reiterate what he said. So he said the countercurrent system is when the blood flow through the vasa recta is opposite to the flow of the filtrate through the loop of Henle. So this allows the filtrate to be exposed to hypotonic blood for reabsorption of water. If they flowed in the same direction, the filtrate would just be in contact with the same blood as it travels. So since they're flowing different ways, there are two different starting points, so they pass at just the right time that the flow goes down their gradient. So like he said, the filtrate is exposed to hypotonic blood. When it's exposed to hypotonic blood, the water is going to flow from the blood into the filtrate. It's basically just the countercurrent. They're keeping the gradient along both of them because they're running anti-parallel to each other. Yeah, Kaplan quick sheets for MCAT is helpful for the high yield stuff. I'm going to look into some other things. Maybe I can find a video or something that better explains countercurrent multiplier. So stay tuned in the GroupMe, and definitely if you have questions about that, come to office hours. But I'll definitely try to find another resource so that we can clarify, because it seems like a couple of people do have questions about that. So I'll get back to you on that as well. I'm going to link you guys to some resources that'll help you with that. And someone asked if we recommend taking other courses. if you think that it's worth it, it's worth it, is basically what my answer is going to be. If you can afford it, I think it's a good idea. They help you, it helps guide your independent studies. Specifically, I'm not too familiar with which courses are the best. I did use Kaplan materials. So I know they are pretty trustworthy. We have some other like adaptive Q banks like UWorld. I know Tim, the CEO, he's doing some sort of promotion for it. So it's like a discount code with that as well. Yeah, and then there are some other like free resources online as well. So like for cars, I know Janelle really likes using Jack Weston. Khan Academy online is improving. I probably wouldn't use Khan Academy for anything too science-based. I think it's pretty good for like the psych and social reviews. Someone in there recommends the Berkeley review as well. If you're looking for the code in the UWorld, you can just shoot a quick message in the group chat, and I believe someone can link you to that there. So yeah, that's really it. We'll see you guys tomorrow for office hours. If you want to stop by, if you have any questions, you can definitely bring them. We'll try our best to help you out. So if you guys have no more questions, then that is it for tonight. Yeah, I hope this was helpful. I'm going to post the PowerPoint. I just have to make a few corrections that I saw throughout. So I'm going to post it in a couple of minutes. We're going to upload it to the OneDrive folder, and then the lecture should also be on YouTube within the next couple of days. Okay. So great job tonight. You guys are awesome. Keep going. And I will see you soon.